Hello dear students, welcome to Biochemistry by Revati. I am Revati, and in today's class we will continue the biological buffers, that is another important buffer system in our body, that is protein buffer system. This also helps regulate pH inside the cells and even in the blood. Now you may get a doubt that we already have phosphate buffer system that regulates pH inside the cells, and an important biological buffer, that is bicarbonate buffer system, that also regulates pH in the blood. Now, what is the need of this protein buffer system? Let's find out what it is actually and how it regulates pH in our body. Let's see what is the need for protein buffer system. First, proteins are the most abundant macromolecules. Since they are present in large amounts, they contribute to what? Buffering capacity. Second, they are versatile buffers. Means they have both acid and basic groups. Acidic means they have carboxyl group. Basic means they have amino group. This makes them showing which nature? Amphoteric nature. Having both the groups. Means here proteins act as both acid and base. So here certain amino acids like histidine, especially effective because of its pKa, that is close to the physiological pH. Means histidine also helps in the buffer capacity. Third, hemoglobin. It is a powerful buffer. We already know that hemoglobin is a protein which is present in the RBC, and it also transports oxygen and carbon dioxide. Now let's see how it helps in buffers in RBCs. This hemoglobin helps the buffer the H plus produced when carbon dioxide is transported. Means here it helps the H plus produced when carbon dioxide is transported. Now let's see the detailed mechanism in further class. And it also complements a bicarbonate buffer system. Means here protein buffer system supports the bicarbonate buffer system by maintaining the stable blood pH. So without hemoglobin buffering, it will be difficult, means the blood pH will fluctuate for every breath. So that much important is the protein buffer system. Now let's see local pH control. We have already seen that phosphate buffer system regulate pH in the cytoplasm, and bicarbonate buffer system regulate pH in the blood. Now what is the importance of this protein buffer system? It regulates pH inside and outside the cells, especially like muscle cells, liver cells, and blood plasma via albumin, and RBCs via hemoglobin. Let's learn what is the protein buffer system. In general, what is actually a buffer? It is a combination of weak acid and its conjugate base. And what is the function or role of a buffer? It helps resist changes in pH. So in protein buffer system, proteins only act as buffers. Why? Because they have an ability to accept or donate protons depending upon the pH of the environment. So what are actually proteins? They are made up of amino acids. So most of the amino acids will have carboxyl groups and amino groups. So here carboxyl group means acid, it donates protons. Basic groups, base accept protons. So because of this behavior, proteins are amphoteric in nature. Why? Because they act as both acid and base depending upon the pH of the environment. Is it clear? Now let's look at the protein buffer system. Means here how proteins act as buffers. How proteins buffer the pH changes. So when the environment is too acidic, when the environment is too acidic means too many H plus ions or protons or hydrogen ions. What will happen? Protein side chains like amino group. Protein side chains like amino group will accept H plus ions to reduce acidity. When the environment is too basic. Basic means what is the volume of H plus ions? Whether they are in increased state or in decreased state? Very low H plus ions. So here protein side chains like carboxyl group will donate H plus ions to increase acidity. So this is all depends upon the pH of the environment. So here certain amino acids like histidine, they are good buffer which are having a pKa value that is approximately 6.8, which is very close to the physiological pH and is making them very effectively in resisting the pH changes. Now let's see one of the most important protein buffers in the body, that is hemoglobin buffer. Let's see how it works. First in tissues. So when the blood reaches the tissues, here tissues will produce carbon dioxide. This carbon dioxide is produced during metabolism, like muscle activity. Now this carbon dioxide enters the RBCs. Inside the RBCs, what will happen? Carbon dioxide reacts with water to form, combines to form what? We have already seen this reaction, carbonic acid. And now this carbonic acid again splits to protons and bicarbonate ions, right? Now, if there is a surge, if there is an increase in the H plus ions in the blood, what will happen? Again, it leads to acidosis. In order to regulate the pH of the blood, what hemoglobin does? This hemoglobin in the form of oxygen. So here hemoglobin binds the extra H plus ions which are present in the blood, and it forms deoxyhemoglobin, and it releases oxygen into the tissues. So actually it is a reversible reaction. It acts both ways depending on the pH of the environment. So here when there is a drop in the pH, when there is a sudden drop in the pH, what this oxyhemoglobin does, it binds extra H plus ions which are present in the blood to form deoxyhemoglobin, and it releases oxygen into the tissues. Is it clear? What will happen in lungs? Already oxygen levels are high, and if the blood reaches lungs, what will happen? Here the oxygen binds to the hemoglobin to form oxyhemoglobin, and this change in the shape of the molecule leads to the result of losing its affinity for H plus ions. For H plus ions. So let's see. Actually, hemoglobin in the form of deoxyhemoglobin. Now, when it combines with oxygen, it forms oxyhemoglobin. So here oxygen combines with hemoglobin and H plus ions are released into the blood plasma. What will happen? So H plus ions are released into the blood plasma. Blood plasma. So when there are low levels of H plus ions in the blood, the deoxyhemoglobin combines with oxygen to form oxyhemoglobin, and H plus ions will be released into the blood plasma by maintaining stable pH, that is around 7.4 blood pH. So now this H plus ions reacts with bicarbonate ions present in the blood plasma to form carbonic acid. This we have already seen. Now this carbonic acid will split into carbon dioxide and water, and now this carbon dioxide will be exhaled from the lungs. So from this what we have come to the conclusion. So when oxygen binds, H plus ions are released. When H plus ions binds, oxygen is released. So it is a part of the Bohr effect. And if more H plus ion binding means oxygen is released into the tissue. When more oxygen binding takes place means H plus ions are released. So here it allows oxygen delivery and also helps acid base balance. So hemoglobin is acting as a gas transporter and also maintains blood pH close to 7.4. Besides hemoglobin, we have other protein buffers also like albumin, which regulates pH in the blood plasma, and some other buffers like enzymes and structural proteins also acts as a buffers which regulate pH. So all of this contribute to the acid base control. Now let's see the summary. First, protein buffer system. It is the most powerful buffer system in the body because it regulates pH in cells, tissues, organs, and even in blood. Second, here the proteins acts as buffers because they have carboxyl and amino group. Third, we have taken hemoglobin as an example and its mechanism. Fourth, they work along with phosphate buffer system and bicarbonate buffer system to maintain what? Homeostasis. That's all for today's video. I hope you have understood the concept clearly. If you have any doubts regarding the concept, please comment on your doubts. And if you found this video helpful, please like, share and subscribe to Biochemistry by Revati for more simple and clear explanations. And in the next video, we will learn the ammonia buffer system and another biological buffer in our body. Until then, keep learning and stay curious. See you in the next class.